Hey guys, I think you can see me. How are you all doing? Ooh, is anyone there? Oh my god, I'm very paranoid that you guys aren't actually here. Can you see me? Looks like it, looks like it. How are you all doing? I see that you are getting slightly scared about UCAPs or whatever it is you're doing, BMATs. Oh, and it was really nice to just read your comments just for a second. So I got here like 10 minutes ago and I was basically being a bit sneaky and looking up what you guys were saying. And it was very nice of you to say that these have been useful so far. I really appreciate it, whoever it was. Panjuni, I appreciate it. Who else said something nice? For one of the last lesson, who said they learned stuff about viruses. Thank you for that. I really appreciate it. I'm glad that you're getting something out of it. I guess that is the main point of all of these sessions, right? Hopefully you go away having learned something. What else have you guys been doing then? So I saw that a load of you have been set a load of biology work. Oh, I bet you're thrilled. Anyone had anything interesting happen recently, or have we all just been bored? Trying to do your EPQ. Tally, what is your EPQ on? Ramadan and biology. Nice. That is a beautiful combination of being hungry and also having to learn. Done an online course for Eton. Ooh, I didn't know Eton did online courses. That is exciting. What else? EPQs, nice. Oh, I'm glad you guys are doing EPQs. What are they on? Does anyone have any like interesting topics? So whether judging history is damaging for society. No, that's fine. I really like history. I'm intrigued. That sounds interesting. Does that mean that you're my voice went? Are you trying to say that if we just let everything happen and then never considered what happened, that would be a good thing? You're in year 11, but in year 12 you're doing an EPQ. Very nice. How technology is used effectively to treat heart disease. That sounds quite fun. Methadone treatment is effective for heroin addiction. Nice, nice. Sounds interesting. Where do you get these topics from? When I did mine, it was really basic. I was like, oh, I'm going to make some aspirin. That was about it. Performance enhancing drugs and sports. Very nice. Oh, this is cool. I regret choosing core maths over EPQ. Ah. Fair enough, fair enough. I don't know very much about Tally, but that does sound interesting. Right, so what we're going to do today, we are going to do some mitosis, if that's not obvious enough to everyone already. So I thought that we'd sort of do a bit of a cell cycle, a bit of mitosis. I don't think there's any meiosis in here, so you can sort of rest easy. No, I'm not going to try and trick you out or anything like that. Not part of the curriculum. Yes, I'm pretty sure they do have to be not part of the curriculum. I think you're right. Mitosis gives me a hug. I'm intrigued. Headache. Headache is less good than hug. Okay, cool. So, just so you know, I have an exciting thing to give away. So you guys are pretty used to my coupon codes, I would imagine. And I'm trying to think of funny things to do for my coupon code. And I don't plan it, so it just whatever happens, happens. But this week, as a bit of a novelty, I'm going to give away one ultimate account for free. So at the end of this session, in fact, no, not at the end of the session, you don't have to do anything. You can start doing this now. Basically, all you need to do is follow SnapRevise on Instagram and then basically tag a picture of you having a fun time doing SnapRevise, like doing this session now or something. So what could you do? I'm looking for creativity. That's what I'm looking for. It probably will be a bit random who we pick, but if you have a nice little post, I'm probably more likely to pick it. So if any of you want a free account, which is worth quite a lot of money, and I don't know how long I'm giving this away for, but it'll be at least a month. I think it's probably a bit more than that, and someone can probably say, one of my people behind the screen can say, yeah, if you want a free account for a bit and you want to come to more sessions, then feel free to tag us in something. Someone won last year, and I can't remember who it was, but I think they were fairly happy. But you'll be getting at least, how many lessons will it be? It's at least one a day. So in fact, that's absolute nonsense. It's at least like, what is it? At least 2 a day. So it's got to end up working out as being like a ridiculous amount of lessons, basically. So yeah, do that. And also look for the coupon code at the end. Okay, will it unlock all subjects? No, it's not going to unlock all the subjects. It'll be biology, because this is biology. You know, biology, like mitosis and biology. Biology, be biology. Yeah, not maths, just biology. Sorry, revision 768, that seemed really sarcastic. I didn't mean to go so sarcastic, but it just happened. I apologize. Right, anyway, so essentially what we're going to do then is we're going to go over some mitosis things, see how you're doing, and then I'll go through some answers. So let's get cracking, eh? So a scientist counted 22 chromosomes in each of the root cells of a xerophytic plant. What is the diploid and haploid number of chromosomes for this species? What do you guys think? This is actually a harder question than you might necessarily think. D's and B's. Oh, I never got to say my interest in little, how long is it going to last for? And is this relevant to all specs? Guys, you've let me down. Okay, mostly B's so far. Couple of D's thrown in. Okay, cool. So yeah, the answer to this one then is obviously between one of those two. It's actually B. Ooh. And I'll tell you why. Essentially, if you can see the chromosomes, the only time that you tend to be able to see them is after they've gone through replication. So think about the cell cycle as having interphase, and then, as you guys all know, PMAT. If we have gone through interphase, you know that partway through interphase, you have a synthesis stage where you end up doubling your amount of DNA. So your chromosomes go from sort of looking like little sausages to looking like little butterflies or little X's or something like that. Okay, so if you can see 22 chromosomes, then that is the absolute maximum number that you're going to see, right? You're going to see 22 as a max. Haploid number is therefore going to be 11. Okay, so in terms of looking at chromosomes, if you can see 22, you're going to be seeing the diploid number, and then the haploid number is just going to be half of that. Which level? Astley, I'm not too sure what you're asking about. Well done for those who forgot that. Yeah, what do you mean which level? I'm very confused. Do you mean like A level or GCSE? So when we mention chromosomes, it tends to mean diploid. Yeah, I would, on the whole, suggest that that's going to be the case. Why is it not D? Because I don't know why you sort of assume that it's going to have double the amount. If you can see 22 chromosomes, then there's 22 chromosomes that are present. You're not, so in mitosis, I guess this is another way of looking at it. In mitosis, right, you're never going to get half the number of chromosomes. So mitosis is where they all line up, right? So I'm going to get eventually all 22 of them lining up down the middle. I know that isn't 22 before anyone says, but essentially you're not going to half it, right? These are never going to be in half. You only half it in meiosis. And I don't know, I just can't see why you'd end up having 11 in your root cell at any time. So the haploid number, the diploid number is going to be 22, and the haploid is going to be going into 11. Okay, so if you're seeing them, if they're all lined up like this, then you're going to have, that's going to be the diploid number. Okay, right. Which row is correct for these diagrams? So we're either looking at, so basically each one is giving you an image, and then you have to say what stage it is. So essentially that's one row. I'll kind of cut the table in half a bit so it would fit. There you go. So what do we reckon is correct here? Most of you are saying D. I'd be intrigued if you guys could tell me what the other ones are. Yeah, you are spot on. So D is 100% showing you anaphase. Someone just said the anaphase, you want to think of like away. So in anaphase, your chromosomes, which started off as your little X's, which are all down the middle, they're getting pulled apart. So you go from looking like that to having half of them that side and half of them this side. What is this picture showing us? This first one. Isn't A interphase? No, A isn't interphase. So during interphase, your chromosomes haven't actually condensed. I can't remember, it's either like uncondensed or decondensed, basically not condensed. So during interphase, you can't really see chromosomes. Okay, so for this one, it looks as if they're trying to move towards the middle. I'd say it's a bit of a mess, if I'm totally honest. I reckon you could argue a couple of things for that. I reckon it's sort of on the border of prophase and metaphase. It looks like they're starting to line up. If I was to compare these all, right, this one looks like it's in more of a straight line than this one. This one looks a bit kind of wonky. So I would say that this one, A, is more likely to be showing you metaphase. It just doesn't look like it's all quite there yet. Whereas C looks a bit more random. It is a bit of a difficult one. I think it's a bit of a, I don't know, maybe they've done this on purpose and they haven't actually shown you a specific phase for those two. What one is B? B is a fairly obvious one. There's not a lot that you could really say. So is C prophase? Yeah, I'd say C is more looking like prophase. It's a bit more random. Yeah, B, I would say, is telophase. So you've got your two sort of nuclei forming, right? So with the series of events or your sequence, right? You got prophase and pro. I never remember if this is Greek or Latin, though someone here must do Greek or Latin or some form of classics. I didn't go to a school to talk about, but pro basically means like first. So in terms of prophase, you know that's the first one. Metaphase, M, I've always thought stands for middle. So if they're lining up in the middle, which they look like they are starting to do in this one over here, that means that it's, yeah, metaphase. A, away, someone said, and then telophase, I've always thought two. So as in you now have two new nuclei which are forming. Yes, so it's not anaphase because they are now at the poles of the cell, right? This one is anaphase. You can tell because of the V-shaped chromosomes as well, right? So because they're looking more V-shaped, that's implying that they've just been broken at the centromere. So your chromosomes all line up like this, right? They have a centromere in the middle. If you break the centromere apart, you end up with one V-shaped chromosome or chromatid, I guess is a proper word. One chromatid there, and you get another chromatid there. What does M say? Middle, if that wasn't very clear. Sorry about my handwriting. Okay, right, next one. So the diagram shows chromosomes at different stages of mitosis. I guess this is going to be a bit of a giveaway about what's going on in that previous question. Looks like we were right. So which shows the correct order of the cell cycle then, out of these? I thought pro was Greek as well. Most are going for B. There seems to be a Harry Potter theme with some of your names. Nice. Yeah, so B is indeed correct. So this is certainly prophase, right? Ooh, it's definitely prophase because there is like no real order. It's just a mess. I know it's not interphase because I can see chromosomes. So this is something which I think confuses quite a lot of students. When we think of our cells and our nucleus, we tend to assume that our chromosomes always look like these little X's. It's just totally not true. So most of the time, if they were to look like anything, they'd look like little sausages. However, because they're not condensed, most of the time, it just looks like a mess. As soon as you can see this sort of structure of this sort of spaghetti-like stuff, that is showing you your individual chromosomes. So that is definitely prophase. So that's got to be first. So I think B is the only one where X is first, actually. So that made that really easy, right? It's obviously fairly obvious as well, obviously fairly obvious, that W is going to be at the end, and that's the only one as well. Condensed means thinner. Condensed means that as opposed to being really long, it starts to wind up in on itself. So your DNA, you've got about 2 meters of DNA in each cell, right? And each of your cells is pretty diddy. So you've got to find a way to get all of that 2 meters worth of DNA into a little cell. The way it does it is you have little proteins called histones. So if I draw like a couple of histones, I'm sure most of you know this. But essentially your DNA wraps around these histones. And essentially the more it wraps around, the more it's going to look like a chromosome. The less it wraps around, the less it's going to look like it and look like a... my face is covering it. Oh, sorry. Essentially what's going to happen though is the more wrapping around there is, the more it's going to look like a chromosome. The less wrapping around, the less. Histones are just little proteins. Okay, so next, in terms of gap phases and stuff like that, some specs do need to know. So OCR like to know about the gap phase. AQA, a little bit, not a lot. Edexcel's a little bit, not a lot. Anyway, so the photomicrograph shows cells in different stages of mitosis. In which order do these stages occur? Have a look. What do you reckon? I will tell you about gap phases in a bit. Don't worry, Kang. So let's have a look then. So in terms of where we're starting, I think that your first cell, which is looking, I guess you've got one which could be an interphase. Some of them are a bit tricky because some of them it looks like it's sort of starting to decondense, right? So if we maybe put some of the middle ones in the order, we might be able to cancel some of them out. So this one here, and it doesn't help that it's a cycle as well. This one here, that could be towards the beginning or the end, right? And this one here could be towards the beginning or the end. But with the other ones, what can we actually tell is happening? What's going to be for, like, surely with 5, 1, and 2, you should be able to tell which ones of those are going to be first. So yeah, 5 is showing you metaphase, so I definitely have 5 there. Out of those next to it, it looks like 1 is showing me anaphase, and then it looks like 2 is showing me telophase. So do I have any of those orders? I do have one of those orders, yeah. So 5, 1, 2 has got to be either B or it's got to be D, right? So it depends if we're going to put 4 first or if we're going to put 3 first. So to me, hang on, wait, does C show that as well? 5, 1, 2. Oh, it does show that, yeah, sorry. Right, so it's got to be one of them, so it's not A. Right, next point then, I've got to work out which one of these I think is first. So do I think that this is going to be the first step, where it looks like that? Or do I think that this is going to be the first step? I think if you look very carefully, it looks to me as if this cell here, oh, maybe not, maybe, is that one connecting with that one and this little faint line in the middle, is that showing you that it's about to undergo cytokinesis and that falls at the end? Or is it trying to suggest to you that they're 2 separate cells? I think this is quite an ambiguous question, which is sad, because I would say that 4 definitely looks like interphase to me, and 3 definitely looks like prophase to me. So I reckon you could get away with possibly saying more than one answer here. And most of you guys are saying C, right? You think they're separate cells. Go on, let's go for C then. I think C is probably the best answer. So in terms of this question, then, we got 4 first. So this is definitely showing you interphase. If they are separate cells, which I guess they are, because if they weren't, I think that this number 4 and this dash would be coming somewhere in the middle, but it isn't. So I would say that 4 is going to be first. Following 4, we've got number 3. So interphase, prophase. Following that, we've got 5, where we've got metaphase going on. Then we've got 1, anaphase. Then we've got finally 2, which is showing you telophase. Okay, so I would go for C. I think C is your best answer, although I do think that's a little bit ambiguous, if I'm totally honest. How do you feel with that? How's it C when it finishes with 2? Because you've got your cells over here which are starting to undergo telophase, or they're in telophase. B is the same answer to C. What do you mean? Oh, it's just starting with 3 first. Yeah, that's true as well, actually, isn't it? I hadn't actually noticed that. So this is just starting it here, isn't it? So 3, 5, 1, 2, 4. What makes this even more stupid is that this is also showing you a cycle, right? What does the mark scheme say? The mark scheme went with C, but it doesn't necessarily mean it's right. So this question could have had a mistake in it, so I wouldn't get too upset with it. So putting 4 or 3 first. No, I would go with 4 first. I think it makes more logical sense to start with interphase, because interphase does come first, doesn't it? Right? Interphase is what's going on most of the time. So if you go back to your cell cycle, right, you've got a tiny bit of mitosis covered here, and it is a cycle. So it is a bit silly to say this, but you are probably starting with interphase, which is going on before mitosis can happen. Interphase has to happen, right? If you went back all the way to the first cell, interphase is going on before mitosis. Oli is blaming the mark scheme. Oli will always blame a mark scheme if I'm getting a little, I don't know, if I'm not 100%. Can I explain the difference between 4 and 3? Yes. So 4 is showing interphase, right? 4 is definitely interphase because I can't see a single structure in there. So 4 has to be interphase because this cell here does not have any chromosomes visible. So that's why I think 4 should be probably going first. I just said should be probably. 4 will go first in our order because 4 is 100% showing interphase, whereas 3 is 100% showing prophase. Okay, because I can see like individual sausage-shaped things, that is implying that this has to be prophase. Now, arguably, 4 could possibly go at the end if these two cells are supposed to be the same. But where this label is pointing specifically at cell number 4 and not in the middle, C has to be correct. I changed my mind. It's got to be C. I agree with the mark scheme, and mark scheme is fine. Why can't we see any organelles? That's a good question. It's been stained so that you can pretty much only see the nuclei. So they've stained it with something like methyl blue, which basically stains chromosomes. A really stupid fact about chromosomes is that chromosomes is the Greek word for colored body, I believe. Right, here's a terrible fact. And essentially, they named it after something which you can't actually see unless you stain it. So you can't normally see the chromosomes unless you stain them. And they decided to define them based on your ability to see them. Pretty dumb. Right, anyway. So in human cells, the tumor suppressor gene, TP53, codes for a protein that interrupts the cell cycle. If there is any damage to the DNA, and prevents copying of the damaged DNA. Sorry. So TP53 is only encoded when there is damage to the DNA. So which stages, A to D, could TP53 interrupt the cell cycle? This question is a lot less ambiguous, you'll be pleased to know. So codes for a protein that interrupts the cell cycle if there is any damage to the DNA. The only place where you're going to get like the DNA changing in any significant way is going to be S. Okay, so synthesis is essentially where you're going to be having changes to the DNA. You're going to have DNA replication. Okay, so it's got to be C. So those of you that asked earlier, essentially your cycle looks like this. So you've got mitosis, which is about 10%. Okay, technically cytokinesis isn't really part of mitosis. That's like a tiny, tiny little bit after. But mitosis is 10%. The rest of it is interphase. And lots of you will know that there is a gap 1 phase, there is a gap 2 phase, and there is the synthesis stage, which comes in the middle of the two. Okay, essentially, the gap 1 stage is where the cell will start to physically grow. You get replication of protein synthesis, you get replication of organelles. Synthesis is specifically where DNA replication goes on, you know, the semi-conservative, all that nonsense. G2, you get a bit more growth. You also get in these G stages just the normal cell functions. So, for instance, if you were a photosynthetic cell, you'd have photosynthesis going on there, you'd have respiration. But in this question, essentially, you get changes to the DNA, you get damage to the DNA, and you get copying of DNA during the synthesis stage. Okay, that's the only time when you have any changes to the DNA. Otherwise, it will be normal. G0 as well. Yes, some of you are correct that essentially the G1 stage, you can sort of go into this other stage called G0, which is only an OCR. Yeah, it's just an OCR thing. And this is basically where everything stops. So lots of cells that just don't do mitosis will just go into this G0 phase. And essentially that's when they'll just carry on doing cell functions forever. So you do get some checkpoints every now and then, but I wasn't really going to talk about them too much. But yeah, you get some like checkpoints such as during metaphase to make sure that all the chromosomes are lined up properly. You get one after synthesis to basically make sure that there aren't any mistakes that have been made of DNA replication. Yes, cool. Okay, so in plants, dividing cells can be found in meristematic tissue. Name 2 parts of a plant where meristematic tissue could be found. So where do we find meristems? Okay, I'm intrigued that some of you guys are using some words that you are using. When you say stem, what does stem mean? Ooh, one person has gone for another answer as well, which I like. Root tip, xylem, cambium, roots and shoots. Right, okay. Time to get out of the habit, which I imagine lots of you are in. So lots of you are saying roots, which is fine. Specifically, you want to say in root tips. Now, lots of you saying stems, I don't think you'd get the mark, right? The other part of a plant that isn't a root is a shoot. Okay, so be careful with this. I wouldn't say stem because it's not like the proper biological word which you should be using. So it's the root tips and it is the shoot tips. Or you could say like the apex or something like that. It's just top of a plant, right? So, I don't know, that's a bit of soil. Here is a bit of a shoot. And then the very tip of that shoot up here, this is where you're going to get your meristematic cells. In terms of a root, which you're going to have numerous of, the root tips, which are going to be down here, you're going to get meristematic cells. Those of you that said in the cambium, that is very good. Who can tell me what a cambium actually is? It's in the middle. So I think the way that I've always considered the cambium is in the shoot, you have this sort of ring, right? And this is the cambium. And on the outside is where you get new phloem cells being produced. And going on the inside, you get xylem cells being produced. Okay, so you have these sets of cells in the middle of your plant, which are called the cambium, and they are also meristematic. Yeah, so between phloem and xylem, exactly. What does meristematic mean? Meristem cells are just stem cells, they're just plant stem cells. Lovely. Okay, so there are a number of stages during cell division. The list, J to N, describes some processes that occur during the division of an animal cell. Okay, so the cell surface membrane is constricted, the nuclear envelope reforms, sister chromatids are pulled apart, the chromosomes condense, the chromosomes move to the equator. Match each letter J to N with the stage of cell division in the list below. First has been done for you. So M, the chromosomes condense. Nice. Metaphase. There is some disparity between what some of you are saying. Metaphase. Lovely. So metaphase, you want to be saying that the chromosomes move to the equator, right? I have something really, really sad to tell you, which is when you get, if any of you do a degree in biology, which I'm sure at least one person, how many of you are here? Hang on. 108 of you are currently here. There's got to be at least 1% of you have got to do a degree in biology. When you go to do biology or biomedicine or biochemistry, you'll learn that the cell cycle actually has another secret phase, which you never get taught about at school, which is called prometaphase. Right? As you might have guessed, it happens between prophase and metaphase. But this is where it's a bit ambiguous because obviously we like to think that the spindle not like attaches to the chromosomes during metaphase, but that's not quite true because they wouldn't be able to get there. So it has to happen during this other stage, which is prometaphase. But anyway, I thought I'd just upset you by telling you one last little thing. Yeah, you learn that in medicine as well. You'd learn it in anything biology related, you'd hear about that. Anyway, so after metaphase, after my chromosomes have moved to the equator, what happens during anaphase? Yeah, it's late prophase. Prometaphase is in an cell. Okay, if it is, which I don't remember that it was, it's very, very little. Okay, so sister chromatids are pulled apart. Very nice. In terms of sister chromatids, are we all fairly happy with what they are? Because people tend to get confused with that term and homologous chromosomes. Yeah, just in case. Essentially, when DNA has copied itself, which it does during synthesis, right? Imagine that I start off with just that. That's what I start off with. During synthesis, it copies itself. So at the moment, that is one chromosome. At the moment, it's one chromosome. After synthesis, I end up getting a structure that looks like that. This is still one chromosome as well, by the way, because you count chromosomes based on the number of centromeres. But this bit here is identical to that bit there, apart from I haven't drawn it identical because I'm a bit of a spanner, but all of these bits are identical to one another. So these are called sister chromatids because all of these bits are fully identical. They've got the exact same alleles. The difference that people get confused about is a homologous chromosome does not contain the same alleles. A homologous chromosome contains the same genes at the same loci, but it will have different alleles within it, right? So your homologous chromosomes, you might, for instance, have different alleles because you got one of them from your mum and you got one of them from your dad, right? But a sister chromatid is the two identical bits of chromosome, right? So those two, which I've drawn blue arrows on, they are sister chromatids, right? This thing over here on the right, that is a homologous chromosome, right? The number of students that don't know that is quite outstanding. Right, lovely. Anyway, so telophase. What happened during telophase? Someone must have said. Crossing over happens between homologous chromosomes, different chromatids, yes. So are two chromatids, one chromosome? Kind of, yes. Yeah, two chromatids are kind of one chromosome, but one chromatid can also act as a chromosome as well. Does it have to all be maternal? No. This is going to be made up. So this maternal chromosome, that is all from your mother, but it's a mix of your grandma and your granddad, right? And when you have meiosis happening, you do swap bits over. So your mixture, your chromosome, so if this is one of the chromosomes that you have, right? It's bits of your mum and bits of your, well, it's bits of your mum and bits of your dad because of crossing over. It's going to be mostly one or the other, but it's bits of your mum and bits of your dad. And this one is going to be bits of your mum and bits of your dad, but it's mostly going to be one. So let's say this one is mostly from your dad and this one is mostly from your mum, then maybe that little chunk down there, that could actually be your mum's, and this little chunk down here, that could actually be your dad's, right? So they can swap alleles over, but on the whole, one of them is from your dad, one is from your mum. Anyway, I got completely sidetracked. We said K is the next one. So during telophase, the nuclear envelope reforms. Agreed. Cytokinesis, the only one we've got left is the cell surface membrane begins to constrict. So M, N, L, K, and J. Lovely. Okay, here is a picture of a cell cycle. So mitosis is part of the cell cycle. Figure 5.2 shows a diagram of the cell cycle. Name one process that occurs during stages G1 and G2. Very good, very straightforward. So protein synthesis happens. Agreed. Cell growth, yep. So interestingly, if you've ever seen a picture of an embryo, they look like little balls of cells, and it starts off with one, and then you end up with two very quickly, then you end up with four very quickly, but it actually stays the same size. It's because these G stages are kind of ignored a bit. So during the first few divisions of embryology, up to about, I think it's like the 64 cell stage, it might be one after that as well, I can't remember. But cell growth is one that normally happens during G1 and G2, but it doesn't always at the very first few divisions during embryology. But definitely those two. Anything else? Someone mentioned, yeah, someone mentioned replication of organelles. Lovely. Does protein synthesis happen in both G1? Yeah, protein synthesis is going to happen during both G1 and G2. See, I would say that you're going to get this happening throughout both, to be honest. So you're preparing for your division. So you're going to get organelles being developed during both. If your spec says that specifically organelle development only happens during G1, I would just learn what it says in your spec or in your book, right? But in reality, you're going to get like a whole mess of things happening in G1 and G2. So why doesn't crossing over happen all the time? Because I don't want to make variation all the time. Right? If I cut my skin, I want my new skin cell to be a pure skin cell. I don't want it to have a mixture of different bits. I want it to just be a skin cell. Okay, so what was that? In a normal body cell, is it just one chromatid that makes up a chromosome? In a normal body cell, right? In a normal cell, if I took a cell from my pancreas, right? Then it's going to have 46 chromosomes in it, but if I took a cell from my pancreas, just a normal cell that wasn't doing mitosis, all of the DNA is just going to be a total mix, right? It's not going to be in chromosomes. It's not going to look like either the sausage shape or the butterfly shape. It's just going to be a mess of chromatin, right? If it was just a normal cell which hadn't gone through synthesis, if it were to turn into a chromosome, it would look more like this one, right? It only ever looks like this cross just before mitosis is going to happen. So this is actually quite rare that your chromosomes ever look like that, right? Most of the time, 90% of the time, it will look like that. Okay, right, so scientists have found the mean length of time spent, this is a really hard question, by the way, so good luck. Scientists have found the mean length of time spent by onion cells in anaphase of mitosis to be 105 minutes. In fact, I'm going to get my highlighter out. So the mean length of time of the cell cycle in an onion cell is 105 minutes. They also found the cell cycle of cells in the onion root shown in figure 1 takes 1080 minutes. So normally the cell cycle is 1080. The average is 105 for anaphase. 32 cells are shown in figure 1, so you don't have to count them, which is fortunate. Use this information and figure 1 to calculate the length of time the cells of this onion root are in anaphase, and then calculate the percentage difference between your answer and the mean length of time found by the scientists. Okay, so what is the what is the percentage difference between the number they found, which is 105 minutes. Ooh, hang on. So the scientist found 105 minutes. What is the mean, the percentage difference between that number and the number that you calculate? So how do I calculate the amount of time spent in anaphase in this picture? I'm going to check the answer for those you give me. Ooh. Some of you who have given me some answers are right. Most of you are wrong though. 32 out of 3, no, not like that. So well done for noticing that there are definitely 3 cells here which are in anaphase. So if I'm looking at anaphase, definitely that one, definitely that one, definitely that one, right? This is what your DNA normally looks like. It's just a mess, right? You can't see anything. It's only when you start to see little sausage shapes like that one that it's in prophase. Yeah, so you're right. 3 of them are in anaphase. I would say this is still looking more T-shapes. In telophase is where you start to see the 2 new nuclei forming within the same cell. Okay, so no, I would say that that one is still looking like anaphase to me. Good, so you do 3 over 32. So if I want to know how many cells, how long the cells are spent in an anaphase, then in this picture, it told me that there were 32 whole cells, right? So out of the 32, I know that 3 of them were in anaphase. Okay, so that will tell me what percentage of them are in anaphase, right? I can then do a nice little trick to work out the amount of time spent in anaphase just based on this one snapshot. So if I know that 3 out of 32 are in anaphase, if I times that number by the length of the entire cell cycle, 1080, that will tell me how many minutes have been spent in anaphase. So 3 over 32 multiplied by 1080 gives me the number 101.25. Okay, so I've got 101.25 in terms of this picture. So the scientists worked out that on average is 105 minutes, right? In this picture, I have found that it's actually 101.25 minutes. So to do the percentage difference, I need to work out what the difference is. So 105 minus 101.25. And then I need to divide that by what I started with. So you do the difference divided by the initial number. So 105 minus 101.25 over 105 times 100 will give me my answer. Okay, so 105 minus 101.25 divided by 105 times 100, and I get 3.6. Yep, 3.57, 3.6. With this question, with questions on the whole like this, there is a bit of a rule that you tend to follow, right? The bit of a rule that you tend to follow is you stick to the same number of decimal places as they have done before you. So you tend to stick to the same decimal places. So arguably, you should get the mark for saying 4%. But in the mark scheme, it actually credits 3.6, 3.57, and 4. So it's a bit of an ambiguous one, and some specs are a bit different on this. But on the whole, you tend to just copy what they've given you, and because there's no decimal places, you tend to just stick to that, right? Arguably as well, some specs would say that you should stick to the same significant figures. And there would be 2 on that one, 3 on that one, and what's that? 3 on that one. So maybe 3 decimal places is fine. So if you did write 3.57, I think that would be totally fine as well. Okay, so describe the appearance and the behavior of chromosomes during mitosis. So during mitosis, what is the behavior and what is the appearance of the chromosomes during mitosis? So what do they look like and what do they do? Cool. So prophase. Maybe I should fill this in. So I've got like appearance there and behavior here. So prophase, what do they look like? What do they look like? Two sister chromatids joined by a centromere. Yeah, arguably they do. They look thick. I would say in prophase that they are visible. I think that is the biggest difference between prophase and interphase, is that the chromosomes are actually visible for the first time. Okay, you can get the mark on this first question by saying 2 sister chromatids joined by a centromere. Okay, in terms of their behavior, you guys were right to say that they are going to condense. Okay, so that is 2 marks of saying that. Metaphase, what happens? Yep, so they line up at the equator. What else? They look like 2 sausages joined together at a point. I like it, Khadija. So they line up at the equator. So behavior. Line up at behavior. And I think we've got to say why they're there. So lined up at the equator. And we want to say they've been pulled by the spindle. Because there's no way that they'd get there if they weren't pulled by the spindle. So the spindle must have actually attached during prophase, right? Or in this pro-metaphase sort of stage. So they line up at the equator. They are pulled by the spindle. In terms of appearance, can we say much else? Joined to metaphase plate by the centromeres. No, you don't have to mention the metaphase plate. Still condensed, uniform line. Yeah, I like that. So yeah, we could say the appearance. Where is the spindle attached? Centromere attached. Yeah. So I think it's still the same as before, but we want to say in appearance, centromere attached to spindle. I think that's good. Okay, anaphase, what do they look like? What's happening? What's their behavior? Centromere splits. They are called chromatids when they've been split, pulled by spindle fibers to opposite poles of the cell, where they are called chromosomes again. Good, V-shaped, right, appearance. I would say V-shaped chromatids. Right, it's a bit ambiguous with biology that when they get separated, they are separated into the 2 chromatids, but because this chromatid here is identical to the one on the right-hand side, a little bit annoyingly, it can also kind of be called a chromosome because it is functionally still a chromosome, right? So I would say that it is separated into 2 chromatids, but from pretty much from telophase onwards, they're called chromosomes again, which is a bit stupid. So V-shaped chromatids, I think that's important. And then we can say they are pulled apart to opposite poles. And then some of your specs like you to say how it works. So pulled apart to opposite poles. One spec, and I honestly can't remember which one's which here. So if whatever one you're familiar with, go with that one. One spec says that the spindles can contract, which is technically true. One spec says they get shorter, which is technically true. And one spec says that they are pulled along by motor proteins. That is also true. So really weirdly, this is like one of the only times in A-level biology where different specs literally just say totally opposite things and they're all true. This is one of those times. So everyone you've heard before, just go with that. So they are pulled apart to opposite poles because the chromosomes contracted, or because the spindle contracted, or because the spindle got shorter, or because a motor protein pulled them there. AQA ignores it. AQA is the only one that I know for a fact says just ignore it. It just doesn't say anything. It just says the spindle pulls apart the chromosomes. AQA has a habit of basically making things a bit more simple. Okay, finally, telophase. There's nothing else we could say there. Stupid pen. Telophase. What do they look like? AQA is the most popular one as well. I think it's about 40% of people do AQA. I think it's about 30%-ish, maybe a bit, maybe it's like 25 or something, or 27 do OCR. Similar with EdXL-A, they're pretty much the same. And EdXL-B is about like 8%. Anyway, daughter chromosomes decondense and unravel. Good. So in terms of their appearance, we could say that they become invisible again, I guess. Become invisible, which sounds a bit stupid. It sounds like you're talking to a toddler there. In terms of appearance, or we could say it becomes longer or something. I guess that's an appearance. And then the reason that it does that, the behavior, is that they will unwind or uncoil, or they will decondense, or whatever word you've used, right? So what will they look like? You won't be able to see them anymore. The chromosomes will sort of disappear. And that's because the chromosomes have uncoiled. It's not invisible. Like, it's like this, right? I can't pick out a chromosome in that picture. I can't see one. I can really clearly see a chromosome there, but in this picture, it's just not condensed enough. It's just really, really unwound. So yeah, even with a microscope, you can't really see them. It's just a long strand of DNA. Okay, right. Let's see how you do on this one. I think I've got a few more actually. I think I put in more questions than I realized. So a student counted the number of cells she observed in each stage of mitosis. Of 200 cells, 6 were in anaphase. One cell cycle takes 16 hours. Calculate how many minutes these cells spend in anaphase. Very nice. So 6 over 200. So you want to work out basically the percentage of cells in that stage. So it's 6 out of 200, which is 3%. And then you're going to times that by 16 hours. But because I want it in minutes, I then need to times it by 60. Okay, so 6 over 200 times by 16 times by 60, and it gives you 28.8. Okay, so 28.8. Arguably, the most significant figures they give you is 16. So arguably the best, well, you should really be turning it into about 29 minutes. But again, 28.8 is still credited. Okay, so be careful with, well, I say be careful, tend to try and stick to what significant figures they've given you, right? Or go thereabouts. 29 is fine. And normally with most questions, they will allow you to not do it all the way anyway. So it's a bit silly. Why do I times it by 16 hours? So essentially what you're doing is you're working out the percentage of cells in anaphase. So 3% of cells are in anaphase. If in this picture, and that was in one picture, right? 3% are in anaphase. if I know that, I'm sort of assuming that from that picture where 3% were in anaphase, that all of the time, 3% of the cells are going to be in anaphase. So if that picture says 3% are in anaphase, then I'm assuming by doing this that 3% of all cells are in anaphase. I'm multiplying it by 16 because I want 3% of 16 hours. That will tell me how much time is being spent in anaphase. Yeah, you can convert it how you like to here. It doesn't really matter in terms of like how you do this equation, but that's how you do it. You could do 60 times 16 first. It totally doesn't matter. Okay, right. I think three more questions. Right. Let's do an applied one. So scientists looking for treatments for cancer are investigating the use of substances called kinesin inhibitors. These inhibitors prevent successful mitosis. Some kinesin inhibitors cause the development of a monopolar spindle in mitosis. The diagram below shows the chromosomes attached to a normal mitotic spindle and to a monopolar mitotic spindle. Chromosomes attached to the normal mitotic spindle on the left, and the monopolar spindle on the right. Suggest why the development of a monopolar mitotic spindle would prevent successful mitosis. So it's going to prevent anaphase from doing what it should be doing. Yes, I agree. So it will stop anaphase. What else will happen? Why is this a bad thing? Why does it prevent successful mitosis? The sister chromatids are not separated. Lovely. Centromere can't split. Yes. DNA is only going to be in one daughter. That's exactly it. Yeah. So if you have this process going on, right? If I'm ultimately going to be making 2 cells, then I'm going to have one which looks like that, which has all of them in, and one which looks like that. Okay, so I'm going to have one cell which has nothing in it, and I'm going to have one which has double what it should have in it, right? They're going to be sausage shaped and they shouldn't be. So it's going to prevent anaphase. I like that. Or you could say separation of chromatids. Oh, sorry, separation of chromosome. No, in fact, chromatids works. Separation of chromatids. And then we want to say something like all chromatids will go to one cell, or all chromosomes will end up in one daughter. The other will have none. That is a problem. Would that be considered a mutation? Yes, it would definitely be considered a mutation. Yeah, non-disjunction mutations is exactly the one as well. Would it have double the number of chromosomes? Arguably, it doesn't have double the number because technically this is only considered one chromosome, which is a bit of a problem. But it definitely has double the amount of genetic material that it should do. But you probably would get a mark saying that you have double the number of chromosomes. Okay, so some scientists investigated the effect of different concentrations of kinesin inhibitor on mitosis of human bone cancer cells grown in a culture. The following table shows their results. Just so you know, because I didn't know this until about 2 or 3 years ago, whenever you get an exam question with like an actual thing going on, like an actual investigation, they actually did this. This is actually real, by the way. Most people don't know this. Anyway, so a student who saw these results concluded that in any future trials of the kinesin inhibitor with people, a concentration of 100 nanomolar per decimeter cubed would be the most appropriate. Do these data support the conclusion? Give a reason. Yep, figures are reality. What do you reckon? Should you use 100 or should you use more or should you use less, or is 100 fine? Anyone? So you're saying you should use 1,000. Why? Good. It should be somewhere between 100 and 1,000. I like it. So yeah, these questions people tend to suck on, if I'm totally honest. 10,000 is too much general side effects, feasibly. Okay, so what you want to see then is a percentage of dividing cancer cells showing the spindle, right? We want as many as possible because it means that they can't do mitosis anymore. So that's rubbish. At 0, we have no effect, so not worth doing. 1, rubbish, not worth doing. 8, pretty bad, not worth doing. I don't want to get rid of only 8% of cancer cells. I want to get rid of as many as possible. This is all right. 100 is all right, but there's still 7% of my cancer cells will still be able to divide, right? 10,000, that's great. That's brilliant. 100,000, sorry, 1,000 is great. 10,000, there's no additional impact of that, so that's pointless. So do the data support the conclusion? No, basically. So there are still some cancers left, or still some cancerous cells left. So you get Marv saying no. Still some cancerous cells left. Yeah, so 7%. You might as well use some data in your answer. So cancer could still spread. All right, it could still be metastatic, which means it's spreading. The best concentration would be somewhere between 100 and 1,000. So between 100 and 1,000. Okay, and essentially what we need, therefore, is we need more research done. So we need more investigations between these values to be carried out. Okay, so that's something else. So best concentration between 100 and 1,000, more research needed. And another point you could make, which would get you a point here, is it says that this is happening to the mitosis of human bone cancer cells in culture. Right? If they're going into human trials, which I believe it says they did, with people, this is in culture, so in a petri dish, not in people. That is a problem, right? Different effects may be seen. Okay, so we don't know the effect on people, which is another problem. Yeah, so I want it to be as close to 100 as possible. 93 isn't quite good enough, and any more than 100, you can't kill more than 100% of cancer cells, so what's the point? That'd just be a waste. Okay, so it's somewhere between those. Right, final question. Kinesin, or whatever it was, inhibitor with a concentration of 10,000 nanomolar per decimeter cubed. In fact, I don't know. They used this to make other concentrations by a series of dilutions with water. Describe how they made 100 centimeters cubed of 1,000 nanomole per decimeter cubed solution of kinesin inhibitor. How do you do that if you start off with 10,000 nanomole of it? For that previous question, basically at 93, it wasn't quite good enough, so you need a little bit more. So we need more research done above 100 and less than 1,000 to see where we can get the effect. And also the other problem was that this experiment was done on someone else, right? It was done in culture, it wasn't done on humans. These maths questions, these are easy, right? This is how you do these maths questions, right? So I want to find out what percent this is of this. Okay, so, and I know I can do this in my head, I know it's 10%, but I'm just doing it on my calculator to make a point. So 1,000 divided by 10,000 equals 10%. So I want a 10% concentration. If I want it to overall be 100 centimeters cubed, then I know that 10% of that I want to be this stuff, and the other 10% I want to be my diluting agent, which is water. So I know that 10% of 100 is 10. So I want 10 centimeters cubed of solution, or of whatever it is, of Ki. And then because I want it to overall add up to 100, that means that the rest of it, the 90 centimeters cubed, has to be water. Right, that's how you do these questions. Work out what percentage the concentration is. This was 10%. Then work out what 10% of this final volume is. That is all you ever need to do. Right, it's much easier than you probably realize. Explain one more time. Yeah, so if it wasn't 100 centimeters cubed and it was 50 centimeters cubed, then I know that 10% of my 50 centimeters cubed needs to be the Ki, and the other, whatever it is, needs to be water. So 10% of 50 is going to be 5. So it's going to be 5 centimeters cubed of Ki. And I know that the rest of it, 50 minus 5 is 45. That is going to be water. Oh, right, I believe that is the end. So very quickly, and I will give you the coupon in a second before I forget. Right, I would imagine if I were you right now, I'd be sat there thinking, God, you know what? That was really useful, and I'd just love to do more of that. I just wish that there was some way that I could be here and I could just do more science. I just wish that, in fact, no, I don't wish. I believe that for me to succeed in life, not just in biology, not just in school, but in life, I believe that for me to succeed, I need to do just a little bit more biology, just a little tinsy wincy bit more biology. You know, I feel like at the moment I'm doing some biology, and my knowledge of biology is up here. But how could I get it to go from here all the way up there, past where the camera can even go? How could I get it up there? How could I get it? Oh, look, I've had an idea. Look, hey, snap revise. Guys, what you need to do, is this regular? I'm going to start doing these stupid things every time, yes. What you should do is, if you have enjoyed this in any way, shape or form, and if you thought, yes, I need more biology lessons or whatever, then look no further than snap revise. Essentially, we have this very, very lovely website which you guys should use. You can even get like a free account for a bit, which will give you some content, but Snap Revise is the best, right? I do web classes not just twice a week as you are currently used to. Oh no, not just 3 times a week, not just 4 times a week. I do web classes blooming loads of times a week. I'm currently doing 3 every day apart from Friday, which for slow maths, 12 hours of time with me a week. So look, tomorrow I'm doing organelles, right? After that, I'm doing sampling practicals, right? Look, it's on the same day, I believe. Tuesday, yeah. Then I'm doing genetic engineering, I'm doing year 13 stuff. Oof, gonna be starting that again soon. Then prokaryotic cells and viruses, I'm doing even more stuff. And if that wasn't enough, blooming heck, look, I do drop-in sessions where I'm available to answer any question you have. So I literally pick up a load of exam questions, go over them. Someone will be like, oh, can you go over the heart? And I'll be like, yes, of course, right? So if you are remotely interested in having any more tuition, because no one's at school, right? There are no teachers teaching you stuff. If you need someone to teach you, I could be that person. And if you thought this was rubbish today, then fair enough. But if you didn't, I'm here to do whatever you want, right? As well as that, look, I've got all these past recordings of me going over random things. Oh, and Biology Book Club, which is a pretty exciting place to be in, if I do say myself. On top of that, on top of all these web classes, let's say, for instance, that you don't know very much about digesting food, for instance, right? What our website does is it basically, we'll play a video, it gives you a little quiz. And you could do this quiz in 1 to 3 minutes. Wouldn't take very long. And you basically go through it. And by the time you've gone through it, and I'm not going to do all of it now because that'd be really painful and boring, it basically tells you what you do and don't know. So for instance, I clearly got a question wrong about the digestion of lipids, so it's popped up here for me. So here are some videos that I should definitely watch, right? Let's say I've watched them and it now goes green because I've watched them and this guy's spoken to me all he wants. Let's say I don't get it still, and I'm really sad. Then essentially, you can post this, and I get a little email pop up that says, I don't get it, and I'm still really sad, and I can then help you, right? So not only do you have videos, not only are there exam questions for when you are done. Oh, look, here is someone who has asked a question. I'm glad I found one. Right, here's someone who said, where can more information be found about chylomicrons? And I have sent them a whole load of stuff about chylomicrons. There are exam questions. So here are some exam questions with some bloke going through it for you. Unfortunately, not me. If you still want more exam questions, there is a quiz that you could do. Right, so you could do another quiz after you've done it and be like, oh, well, I didn't know that bile salts were stored in the liver before. And apparently they're not. They're stored in the gallbladder because I'm an absolute moron. They're made in the liver. That's embarrassing. Let's do another one. Say I didn't know which of the following enzymes break down lipids. So I didn't know it was lipase before. And now I do. Oh, yeah, I got it. right? If that's still not enough questions, then look, you can go through all of these questions, right? And let's say you're choosing a free structure shown in a diagram and explain how they link to their function. Click view solution and bam, there is. Look at many there as well. Look at that. That could all be yours at your fingertips. It could all be yours, right? Anyway, loads of them. I'm not going to carry on badgering you for much longer. And there is also a revision guide, just in case you still don't have enough and you want to print this out and stick it up in your bedroom and make it A10 or something, or A1, make it huge, right? You can put all of that everywhere and it covers like all exam tips as well. Okay, if I have not sold you Snap Revise, which I do understand because I have been talking forever and it's also quite annoying, I agree, then have a look at some more of our YouTube videos. So these are all free and you can basically set a reminder so that you can make sure you don't miss them. So someone mentioned at the start that they nearly missed one, right? If that is you and you've done mitosis now, say you want to do Newton's law tomorrow, then set a reminder and you can go along. And it's free and it will carry on being free and we can carry on doing these YouTube sessions for a little while, right? Probably not forever, but we will do them for a while. If you want to go to membranes and transport, click set a reminder and you can go. Otherwise, I will very quickly show you the voucher so that you guys can do whatever. And if you have any questions, feel free to ask me any questions now. By the way, here is the voucher. Mitosis 10, it expires tonight. Don't forget as well, I've given you £10 off. Don't forget, if you tag us on Instagram, and if you do something funny, I will be more likely to pick you. You might be able to get a free account, right? The account, the ultimate package is £50 a month. Just so you know, it's £50 for the month. But what you got to consider is you also get like 12 hours of me per week. So what's that, 24, 48 hours of me per month. So you'd be paying like £1 per lesson basically, which is ridiculous because I used to charge 40 quid when I was a tutor per session. So if you want to use a code, use a code. If you want to tag us on Instagram, maybe do that first, right? Maybe do that first. See if you get your free account, and then if you don't, do that. And I think we're going to pick out the lucky person on Friday. So maybe if you can't wait, then do this. And then, yeah, maybe post something on Instagram. How many people are on right now? 83 people are currently watching. So you have a 1 in 83 chance of winning. And I like those odds. Like the odds of winning the lottery. Bear with me. Forgetting, oh, that's not right. It says 1 in 10.3 to get, oh no, that's 2 numbers. Okay, hang on. If you wanted to get the winning 6 numbers, it's 1 in 45 million. So just think you are 45 million divided by like 80, you are 562,500 times more likely to win this today than you are to win the lottery. There's some good stats for you that I'd like. Anyway, right, I'm getting distracted. Does anyone have any questions very quickly that I can answer? No worries, Sammy. I'm glad you enjoyed it if you are still here. How much of that is available on the free trial? So on the free trial, you'll get like, I think a few quizzes and you'll get the first few bits of like A-level biology. So maybe like the first 3 or 4 topics, I believe. You don't get the online videos of me, unfortunately. Have done. Nice. Are centrioles and motor proteins? Centrioles are the things that make the spindle. Although there are some people that don't believe that because you can actually destroy the centrioles and you sometimes get spindle fibers forming. So motor proteins, if you want to Google something really cool or YouTube it, go on YouTube and type in kinesin. They are the coolest little proteins. They basically they have little legs and they carry things along. Yeah, I don't think I've actually ever bought a lottery ticket. So that was me having to Google that stat a second ago. You can't understand it. You're going. Okay. No worries, guys. I'm glad you've enjoyed it. So what happens to the centrioles when the chromosomes split into chromatids? Do the centromeres actually split? Yeah, the centromere splits in half. The centrioles start to break down. Tag us on Instagram. Yeah, not specifically me. If you can find me, don't tag me. Tag Snap Revise. Yeah, tag Snap Revise. If you have a private account and we can't see that you've tagged us or something, you might have to send us a direct message, but I think it should work. Awesome. Right, guys, have a really lovely rest of your day then. I've already gone like 10 minutes over, which I didn't mean to. Have a lovely day and I will catch you on Wednesday. We'll do that. Right. See you guys a little later. Adios.